These healthy tobacco plants are about to get sick. They're part of a new push to improve diagnostic tests for the Ebola virus. According to the World Health Organization, the Ebola outbreak that started in West Africa in 2014 has sickened nearly 30,000 people, killing about a third of them. These plants aren't being infected with Ebola itself, but with the tobacco mosaic virus.
In my lab, we use plant viruses as a nanotechnology, as a platform technology to develop novel tools to diagnose disease.
With support from the National Science Foundation, biomedical engineer Nicole Steinmetz and a team at Case Western Reserve University are using tobacco mosaic virus, or TMV, to make what's called a positive control for Ebola tests. The goal is to reduce the number of false negative results. In the past, Ebola diagnostic tests, or assays, have been considered reliable only up to a point. Most tests screen for Ebola genes extracted from a blood sample. The Ebola virus doesn't use DNA to store its genetic code, but a chemical cousin called RNA. And extracted RNA degrades easily. Just one little mistake at the start of a test can ruin the whole thing.
You never know if they might have messed something up. Somebody came and talked to them, or they got distracted in some other way, they dropped a sample or whatever.
Steinmetz partners with breast cancer researcher Ruth Keri. Neither has worked with Ebola before, but both are experts at developing diagnostic tests for diseases.
You always need to have a positive control. So if you're working with RNA, it's just that much more important to do that.
In the lab, they're inserting scrambled up Ebola RNA into the tobacco mosaic virus genome. That's the positive control. TMV on the outside, a snippet of one harmless Ebola gene on the inside. They'll mix that together with a blood sample at the start of a test. So every assay, whether it reads positive or negative for the full-blown Ebola virus, should at least detect the control.
So if my positive control doesn't show up at the end of the assay, then that doesn't mean that the patient was tested Ebola virus negative. That just means that the assay didn't work.
They are also working to make a simple diagnostic test for use in the field, with no need for the sophisticated equipment they have to use now.
We start with a non-colored or slightly purple-colored solution that, if Ebola virus is present, or in this case our positive control, turns into a blue coloration.
Saving lives by accurately diagnosing infected individuals before they spread Ebola. That is most definitely a positive development.